Actually, let's step back and define what are Xenobots.
So what we've done is to take some skin cells off of an early frog embryo and basically ask about their plasticity. If we give you a chance to sort of reboot your multicellularity in a different context, what would you do? Because what you might assume by the thing about embryogenesis is that it's super reliable, right? It's very robust. And that really obscures some of its most interesting features. We get used to it. We get used to the fact that acorns make oak trees and frog eggs make frogs, and we say, well, what else is it going to make? That's what that's what it makes. That's a standard story. But the reality is, and so you look at these skin cells and you say, well, what do they know how to do? Well, they know how to be a passive, boring, two-dimensional outer layer, keeping the bacteria from getting into the embryo. That's what they know how to do. Well, it turns out that if you take these skin cells and you remove the rest of the embryo, so you remove all of the rest of the cells, and you say, well, you're by yourself now. What do you want to do? So what they do is they form this little this motile little creature that runs around the dish. They have all kinds of incredible capacities. They navigate through mazes. They have various behaviors that they do both independently and together. They have basically they implement von Neumann's dream of self-replication, because if you sprinkle a bunch of loose cells into the dish, what they do is they run around, they collect those cells into little piles, they sort of mush them together until those little piles become the next generation of Xenobots. So you've got this machine that builds copies of itself from loose material in its environment.